You know, finding a new medicine can take billions of dollars and more than a decade. It's a huge undertaking. But what if AI could change all of that? Today, we're going to dive into a research paper that introduces a new and frankly incredibly efficient AI approach. It's all about working smarter, not just harder, to discover the cures of tomorrow.
I mean, this is the multi-billion-dollar question, isn't it? The whole process of finding new drugs is notoriously slow, it's super expensive, and it is just full of dead ends. So the real challenge for scientists today is figuring out how to make this whole pipeline faster, cheaper, and, ultimately more successful. This is exactly where AI promises to be a total game changer. All right, so here's how we're going to break this down. First, we'll look at the big picture problem. Then we'll dive into the specific technology they're using and the challenges that come with it. After that, we'll check out the clever new solution, see the impressive results, and finally, we'll wrap up with why this new approach is such a massive deal.
Okay, let's kick things off with Part 1, the Drug Discovery Challenge. And it is a challenge. Finding a new drug is a truly monumental task. We're talking over a decade, billions of dollars, and a heartbreakingly high number of potential compounds that just fail along the way.
This is where AI steps in as a really powerful ally. Just imagine being able to test 1 million potential drug compounds without ever touching a single test tube. That's the promise here. These computational models can simulate how cells might react, letting researchers sift through this vast ocean of possibilities and zero in on only the most promising candidates. It saves an unbelievable amount of time and money.
All right, moving on to Part 2, let's see how this actually works. Because to teach an AI about drugs, you have to show it what they do. And one of the most powerful ways to do that is a technique called cell painting. But as you'll see, it comes with a major catch.
So cell painting, it's pretty much exactly what it sounds like. Scientists use these different colored fluorescent dyes that literally attach to different parts of a cell, the nucleus, the mitochondria, you name it. And what you get is this incredibly rich, super detailed image that gives us a snapshot of the cell's health and how it's responding to a drug. For an AI, these images are an absolute goldmine of information.
Okay, but here's where we meet the villain of our story, batch effects. Think of it like this. Say you're trying to identify your friend in a dozen photos. Simple, right? But what if every single photo is taken with a different camera, under different lighting, maybe even at a different time of day? Your friend is in every picture, but all those variations make it really hard to focus on their actual, consistent features. Well, that's exactly what batch effects do to cell data. They introduce all this experimental noise that can totally confuse an AI.
So this noise problem really throws up 3 massive hurdles for the AI we have right now. First, they really struggle to separate the true drug effect from all those batch effect distortions. Second, to even try to do that, they often need these huge, super complex models with literally billions of parameters. You can think of those as the internal knobs the AI has to tune. And third, these giant models need to be fed enormous amounts of data. The whole thing just becomes expensive, slow, and honestly out of reach for a lot of labs.
Which, of course, brings us to Part 3 and the hero of this paper, a new, much smarter AI framework called the Crosswell-Aligned Masked Siamese Network, or let's just call it CWA-MSN for short.
You know, the goal here is just beautifully simple. This new model is designed from the ground up to do one thing, and do it really, really well. Find the true biological signal, the actual effect of the drug, and ignore all that distracting experimental noise from the batch effects.
And the secret sauce? Well, it's actually right there in the name. The core innovation, the big aha idea, is something these researchers call Crosswell alignment.
Okay, the logic here is, it's just brilliant. The AI is specifically trained to compare 2 different samples, and it's told 2 crucial facts up front. Number 1, both of these samples were treated with the exact same drug. And number 2, they came from totally different experimental batches, or wells, which means they are definitely full of that random noise we talked about. So the AI's only job is to find what's identical between them. By forcing it to do this, to align these 2 signals, it naturally learns to ignore all the noise that's different between the batches and focuses only on the consistent underlying signature of the drug itself. It's just a really clever way to teach the model what to pay attention to.
So that all sounds great in theory, right? But does it actually work? Let's get to Part 4 and look at the head-to-head results, because the researchers put their new model up against the reigning champions in the field.
Okay, so here's the matchup. In one corner, we have our new lightweight contender, CWA-MSN. And in the other corner, we've got the heavyweights, OpenPhenom and CellCLIP.
Two of the absolute best, state-of-the-art models out there.
And wow, the results are pretty stunning. On a key benchmark test, CWA-MSN beat CellCLIP by a solid 9%. But look at this, it beat the other heavyweight, OpenPhenom, by a massive 29%. I mean, to be clear, that's not just a little bump, that is a huge leap in performance.
But here's the thing, as impressive as those numbers are, they don't even tell the whole story. Because what's truly revolutionary here isn't just that it won, it's how it won.
Okay, first let's talk about the data. To get that 29% better score, CWA-MSN was trained on just 200,000 images. Its competitor, OpenPhenom, needed a whopping 2.2 million. That is over 10 times less data to get a significantly better result. Let that sink in.
And if you thought that was crazy, wait till you see the difference in the model size. Our model, CWA-MSN, has about 22 million parameters. CellCLIP, the other big model it beat, has nearly 1.5 billion. That means CWA-MSN is 67 times smaller and less complex, and it still performed better. This is just a massive, massive breakthrough in efficiency.
And that brings us right to our final section. Why does any of this matter? Why is this efficiency so important? What's the real-world impact of being smaller, faster, and way less data hungry?
Look, the power of this kind of efficiency, it's completely transformative. It means you don't need a massive, perfectly curated dataset to get going. It means the computing power you need to train and run these models is drastically lower, which slashes costs. And ultimately, what that does is it makes these cutting-edge AI tools accessible to way more scientists and research labs, not just the ones with gigantic budgets. It basically democratizes the whole playing field and can accelerate discovery for everyone.
You know, the researchers themselves really say it best. They've created a simple and effective method that just works incredibly well, even when you don't have a mountain of data or a supercomputer to run it on.
And that really leaves us with one final big question. This paper proves that in the race for discovery, the smartest approach isn't always the biggest one. And it really makes you wonder, what other massive scientific challenges, even beyond drug discovery, are just sitting there, waiting for a solution that's a little more clever, a little more elegant, and a lot more efficient?